Welcome to the second session of the revision video. This is lesson 5, complex patterns of inheritance. So what does that mean? It's a bit more complex than this normal one. So what are examples of these complex inheritance patterns? We have incomplete dominance, we have codominance, we have multiple alleles, epistases, sex-linked traits, polygenic traits. Gamete, as I said to you before, is a mature sex cell, either the sperm or the egg, with a haploid number of chromosomes. For example, the number of chromosomes which a normal Homo sapien would have is 46. But when we say haploid, we mean 46 divided by 2, which is 23. That's where we got the word haploid from. It means half of the number. So in incomplete dominance, in some organisms, heterozygous individuals would display the dominant phenotype. This is what we know in heterozygous individuals. In heterozygous individuals, we have some dominant, we have some recessive. But in incomplete dominance, the heterozygous phenotype is an intermediate. It's not going to be dominant, it's not going to be recessive, it's going to be in between. So a good example is when you have red flower, you breed with a white flower, a plant with a red flower and a plant with a white flower. So instead of having a red or a white, you got a combination of the coloration. Then you have a pink flower. The pink flower is a phenotype, as you know, but the genotype is the red and the white combined together. So if you decide to cross these hybrid, this is the parent, this is parent. So this is F1. So you want to produce F2 from them, you have to cross these two, as you did here. You cross this with this, and this, and this. So what we had is one red, one pink, and one white. So that is to say you have ratio 1 to 2 to 1. So the phenotype will give you ratio 1 to 2 to 1. What about if you want to look at the genotype? The genotype would also be almost the same. Because why? Because the genotype from this is CR red, and this is white, this is pink. So it's a combination of the two. So you would have, this would be RR, and this would be W and R. And this would be W and R, and this would be WW. So this is how the genotype would look like. So it's going to be ratio 1 to 2 to 1 for the genotype term. Now, in contrast to the incomplete dominance, we have another one called codominance. In codominance, where the dominant and recessive alleles are expressed in the heterozygous individuals. So the meaning is in a kind of flower where you have a red flower, a plant with a red flower, then you cross this flower with another flower, which is yellow flower. So what you would have is an offspring who would be having some yellow and some red. So this is the kind of flower you would have. You would have a flower which would gonna combine both the traits from both parents. So this is an example of codominance. So codominance is studied a lot when it comes to sickle cell. Sickle cell is a disease whereby the red blood cell, instead of having a round shape, become a sickle shape like this. So it doesn't allow the person to carry enough oxygen. So it makes the person lose oxygen and the person gets weak. So changes in the hemoglobin cause red blood cell to become sickled. The hemoglobin, which is a protein on the surface, which controls the production of the cell, is being sickled. So people who are heterozygous for the traits have both the normal and the sickle shape. So this is what we say is codominance. Let's say a person who is normal is having SS, and a person who is sickle is got this. So if you decide to cross these two, what you will get is you will get some heterozygous and some homozygous. So any person who is heterozygous for the sickle cell, because sickle cell, as we know, is a recessive disorder, so you need to inherit both lowercase s before you can have it. But we have people who are heterozygous to it. So the people who are heterozygous for the trait, they have it normal, they're gonna have it uppercase S and lowercase s. So these heterozygous people, they are the ones who we say they are having codominance. They have both the low on both the dominant and the recessive in them. So in sickle cell makes it easy. I mean, malaria and sickle cell have something to do in common. What does it mean? Those who are heterozygous, that is to say, let's say we have one parent and another parent like this. So you take one allele, you take second allele. If you cross these two parents, you would see a hybrid this. So when you have any offspring with this allele is heterozygous. So the meaning is got high resistance to malaria. Why? Because the sickle chip doesn't allow the malaria to be able to grow properly. So they would not be able to survive in this person. So the people who are heterozygous to sickle cell, they don't usually have malaria. So the death rate due to malaria is lower because the sickle trait is higher. So they don't usually die from malaria because they have a higher sickle cell. So the meninase, they will live longer and pass the sickle cell to their own offsprings. So this is the reason why sickle cell is very, very common in places where malaria is also very common. So another good example of the complex patterns is multiple alleles. We have some traits in the body which is controlled by multiple alleles. A good example is the blood group. Some form of inheritance are determined by more than two alleles, which say multiple alleles. The ABO blood group has three forms of alleles. Sometimes we call them AB markers. So these AB markers, they're very effective. So whenever you find, these are the possible gamete from female parents, all of this, and these are the possible gamete from the male parent. So I would explain them to you. A person who's got marker A on the surface of a red blood cell would have this. This person has got a marker B. This one has no marker. The same thing here. So when you cross these two, you have two A markers, one A and B marker. One A marker, no second marker. So AB markers, BB markers, B and no marker. A marker, no marker. B marker, no marker. No markers at all. So what would happen is every time you find one A or double A, at least one A or double A is going to be having a blood group A. Every time you have one A and B, you're going to have AB blood group. But whenever you have BB or B without nothing, you're going to have a B blood group. And when you have nothing at all, like this, you have an O blood group, meaning the person does not have any markers on the red blood cell. So it's going to have a blood type O. And there is something called the rhesus factor, which if the person is got rhesus factor dominant, is going to have a plus. It's going to be either A, B plus, A plus, O plus. If it doesn't have it, it's going to be AB negative, A negative, B negative, O negative. So this is how we do it. So another example of multiple alleles is a coat color of rabbits. So multiple alleles can demonstrate a hierarchy of dominance. The meaning is, although we have multiple alleles, but the hierarchy of these alleles are not the same. Some have a higher hierarchy than the other ones. For example, in rabbits, we have 4 alleles that code for the coat color. We have these, we have these, we have these, and we have these. So the hierarchy of these is more than all of them. But the hierarchy of days is more than these two. The hierarchy of days is more than this, and the hierarchy of days is more than this. Yani hierarchy means daraja. So the presence of multiple alleles increases the possibility, the possible number of genotypes and phenotypes. The meaning is when the number of alleles responsible for a specific trait is more, there would be a higher possibility of genotypes and phenotypes of the individual. The other one is epistasis. Epistasis is an interaction in which one allele hides the effect of another allele. One allele would shade off, would cover up the effect of the other one. It's not that it actually dominates it. It's there, but it's actually covering up the effects of it. A good example of this is seen in the coat color of Labrador retrievers. So the dominant allele E is the one that determines whether the coat would be dark pigment. So this is the one responsible for the melanin level. There's a second allele B, which determines how dark the coat would be. When the dog has a recessive EE, so when the dog has these alleles, the coat will be yellow because the EE allele masks the effect of the B. So the E would mask the effect of the B. So the meaning is if E is not there, then the coat will be darker. But if this is there, it's going to mask the effect of the B. It becomes yellowish in color. So this is what we mean by epistasis. There's another thing called dosage compensation. As you know, in the sex cell, a male has one X and one Y chromosome. These are the sex chromosomes, while the female has XX. So the summary of dosage compensation is during gamete formation, during cell division, the female would switch off one of its X's so that it could compensate for one which a male has. So they will both have 1 and 1. Because the X chromosome is the one that carries the variety of genes that are necessary for development of both the males and the females. The Y chromosomes, they mainly have genes that relate the development of the male characteristics. So that's the main function of the Y chromosome. They just form the male characteristics. In the female, one X chromosome is inactivated. It's been switched off or turned off. So we call this dosage compensation. So the dosage compensation means in activation of X allele. So which X stops working, it's now, we cannot predict. It could be this, it could be this. It's a random thing. So this happens in all mammals. So we want to show an example of the chromosome inactivation. So this is the coat color in the calico cats. So the calico cats, they usually cause a random inactivation of a particular X chromosome in them. There are some coloration which is found on the X chromosomes. So the color that results depends on the X chromosome that has been activated. Orange patches an activation of X chromosomes that carries allele for black fur. For example, this is X chromosome, and this is the allele for the black fur, and this is allele for the orange fur. So during cell division, what happens is they divide into two, and one of the X chromosomes get inactivated. So this is inactivated, the one carrying the black fur. So what happens is the cat would have an orange fur. And all the other way around, if the allele responsible for the orange fur is being inactivated, the black would have a black fur. So this is what we call dosage compensation. Bar bodies. Bar bodies are actually being noticed when the chromosomes of the inactivated X chromosome has been observed under the microscope. So the cells that has these X chromosomes was been found that the dark stain substance, which is the inactivated X chromosomes, was found in the inactivated one. So the X chromosome, which is not activated, they usually have a dark stain substance inside them. These substances are found to be bond bodies. So how do we predict disorders? Due to the sex-linked traits. This is a bit complex. We need to pay attention. Traits controlled by genes located on X chromosomes are called the X or the sex-linked traits. They also call the X-linked traits because male only have one copy of X chromosomes. They are more affected by the recessive X-linked traits. Listen again, because males only they only have one X chromosomes, so they have more effect from every recessive X-linked trait because they don't have a second X to cover up for it. But a female can have a second X with a dominant allele. But a male, let's say, for example, this allele is causing a disease, which is recessive X-linked trait. So if a male has these, the Y doesn't carry anything. So the male would easily have the disease. Because what? Because it doesn't have a second thing to mask it. But a female would not have it because the uppercase B or uppercase A would mask the effect of this. So it's going to be fine. So some traits that are located on autosome may appear to be sex-linked, even though they're not. So this is a bit related to linking genes on chromosomes. So genes are very close to each other. They link together. Genes that are very close together, they get inherited together. But genes that are far away from each other, they cross over. So this is something we did in the other lesson, but it's a bit linked to this. So make sure you understand the relationship between them. So some traits are located on autosome may appear to be sex-linked. So because they are located on the same autosome, so they may appear to be sex-linked, but they're not. They just that they've been on the same chromosome. So now sex-linked traits. Red-grain color blindness is a good example of. Now let's take a look at a good study about this. When a person is red-grain blind, what would happen is recessive X-linked traits, mothers are the carriers. The females can be colorblind only if they inherit the recessive allele from both parents. The recessive allele, let's say, most of the time, the allele responsible for the colorblind is a recessive allele. It could be B, it could be P, it could be N, it could be U, it could be L, it could be wherever. We always use any letter to represent it. So the number one is X uppercase B. The red-green colorblind is X lowercase b. And the Y chromosome is this. So what happened is, this is a male, this is a man. The man has a normal allele, but this is a female. He has one normal allele and one red color blindness. So what would happen is, how do you know when you cross this person will be normal, this person would also be normal. This person would be normal because it's got one normal to mask it, but this person would have it. He would have it because there's no second to mask it. And this is surely a male. This is also a male. These two are female. So be very, very careful when it comes to this. So when you pay attention, you will see because a female has got two X's, it could have a dominant and normal one to mask the effect of, because you have to inherit two recessive alleles before you can have it as a female. But a male wouldn't have it if it has only one. So this is why it's very, very dangerous. So another one is hemophilia. Hemophilia is a disease when the blood refuses to clot on time. So this can cause a problem. can cause people to lose their life, bleeding to death. So there is a sex-linked trait here of Queen Victoria, is the Queen Victoria pedigree. So you will say, you can study this yourself. This is Edward, Duke of Kent, and this is Victoria, Princess of Saxe-Coburg. So these two people seems to be fine, probably the carriers. So, or one of them a carrier. So what happened is we just noticed that Queen Victoria of England became a carrier. Mind you, we're dealing with a recessive X-linked trait. So once you have one allele, once you need to have two alleles to have the disease. So this person is a carrier. So, but what happened is Leopold, Duke of Albany, had the disease. So then he got transferred to Alice, became a carrier. Then Rupert also had the disease. So you will see the normal females are the one who the yellow shaded, not the yellow one. So the normal female. So the normal male is the square. So the carrier is the one half shaded. The possible carrier female, which is not known, is possible carrier. This is it. So this one is affected male. So affected male is the square, which is completely shaded. So now let's talk about the polygenic traits. Polygenic traits arise from the interaction of multiple pair of genes. So polygenic traits means that we have multiple genes responsible for the trait. So a good example is the skin color, the height, or the eye color. So then this is the number of dominant alleles. This is the frequency. So the meaning is the number of dominant alleles you have, the more melanin level you're going to have. This person doesn't have any dominant and it's got number of dominant is 0. It's got a lighter skin. These people have all of it. They have all dominant. They have the darker skin, more melanin. But most of us fall in between this. We fall in between these. So this is why it's called a dumbbell shape. So most people will fall in between these where we have like 2, 3, and 4. So environmental influences can also have effect on the kind of the way people look like. So the variation and distribution of traits in the population depends on environmental factors, because environmental factors affect the expression of some traits. They affect the probability of those traits appearing in the population. A good example is the sunlight and water. Without enough sunlight, most plants will not produce flowers. Insufficient water causes plants to drop their leaves. Temperature, most organisms experience phenotypic changes with extreme heat condition. So the twin study was actually being used by the scientists to separate genetic contribution from environmental contribution. Traits that appear frequently in identical twins, they are least partially controlled by heredity. So traits that appear frequently in identical twins, they are least partially controlled by heredity. Traits expressed differently in them, they're strongly influenced by the environment. So that's the summary. So these are some examples which occur when heterozygous phenotype is an intermediate phenotype within two. So when it's an intermediate, it's going to be incomplete dominance. But I think the answer here actually was wrong. So you should be mindful of it when you're answering these questions. Do not be eager to answer the question without taking your time. So this says incomplete dominance, but that's the answer. So the answer is incomplete dominance, which is an example of how traits can be influenced by the environment. So how can traits be influenced by the environment? Is it the coat color, reading ability, flower color, human blood type? What do you think? So the answer is B. Which two terms best describe the inheritance of human blood types? So human blood types, it could be as a result of what? Of multiple alleles. And is it incomplete or complete dominance? We know about multiple alleles, but is it complete or incomplete? Is codominance. So which is true about sex-linked traits? They never seen in female, they carried they pass on from mother to son. So what is episode says when one G allele masks the all effect of the other one. So intermediate concentrate and activation hiding one but not another one. Okay, boys, I wish you all the best. Have fun. Thank you very much.